Okay, welcome to the lesson. So today we'll be doing introduction to bacteriology, just an overview for you to understand. So as we know, we have branches of microbiology. You did virology. There is bacteriology, which is the study of bacteria. There's mycology, which is the study of fungi, which you're going to do. There's also parasitology, which is the study of parasites, and immunology. So immunology, it's not really a branch of microbiology, but since it has a great relation, it's usually put under the branches of microbiology. And then we have something called mycobacteriology. Basically, what this means is study of species such like mycobacteria. You know, mycobacteria, as you'll see, they don't actually fall under certain bacteria. We're going to see the reasons. But yes, basically the study of mycobacterium, which is TB. Okay. Okay, so we'll do the general characteristics of bacteria. We have number one, bacteria are prokaryotic microorganisms that do not contain chlorophyll. So chlorophyll are found in plants. So we have eukaryotic and prokaryotic. Who knows the difference between eukaryotic and prokaryotic?
I think eukaryotic have organelles, whereas in prokaryotic do not have organelles.
Not really. Basically, we are talking about the nucleus. So prokaryotic are cells without a nucleus, and then eukaryotic are cells that contain a nucleus, okay? Then you can talk about different organelles. Yes, that can also work, but the main usually differentiate with the nucleus, okay? And then here is the differences.
What of things like endoplasmic?
Yes?
I was saying like the lack of endoplasmic reticulum and Golgi bodies.
Yes. So nucleus, as I said, the main defining feature, they are absent in prokaryotes. Nucleolus, deoxyribonucleic acid, chromosome, it only has one circular one. Mitotic division is absent. In eukaryotes, they are all present. Cytoplasm, cytoplasmic streaming, pinocytosis, mitochondria, all these organelles, they are absent in prokaryotes, but present in eukaryotes. And then chemical composition, sterols, moramic acid, absent in prokaryotes, present in eukaryotes, moramic acid present here, absent. We're going to see the exact bacteria that has moramic acid, because not all of them have. And then the size of a bacteria, okay. So as you can see, a bacteria is very, very small. We measure it using micrometer, okay, mu meters. This is 10 power negative 6 of a meter, 1,000th of a millimeter. Okay, so you can see it's a very small thing. You can't see with your naked eyes. Okay. Then we have different shapes of bacteria. These are usually the main classifications. We have coccus. Coccus, as you can see, they are round. Rod, okay, we call them bacilli. And then we have spiral shapes. So for the coccus, we have diplococcus when they are two or in pairs. We have streptococcus, as you can see, they're in chains. Tetrad, if there are 4, then staphylococcus, as you can see, they're in clusters. Then rod, also known as bacilli, this is a single rod. Diplobacilli, if they're in pairs. Streptobacilli, if they're in a chain. Okay, then coccobacilli. Coco, so coco means the round, then bacillus, so it looks like a lactobacillus. It's a round and it's also rod-shaped. And then we have spiral. So spiral, as you can see, it's forming like a spiral shape. Then we have spirochete. Okay. And then we have vibrio. So vibrio, we call it comma-shaped. Okay. If you can see here. Then microscopy. So optical or light microscopy. So you'll appreciate this more when you start your lab sessions. I don't know if you've started, but you'll be able to see these bacteria under a light microscope. So there are different ways. We have the light microscope. We also have dark field microscope. So this is used to observe spirochetes. Okay. So spirochetes are like Treponema pallidum, the one that causes syphilis. Okay. They usually produce a bright image. Yeah. This white in a black background or a dark background. This is dark microscopy. For microscopy, we have phase contrast microscope. This is basically used to differentiate organelles. And you see, so they'll appear in different shapes and colors. Okay. So retardation by a fraction of a wavelength of rays of light that pass through the different cell organelles results phase difference between the two types of rays. Okay. In this phase contrast microscope, phase differences are converted into difference in intensity of light, producing light and dark contrast of the image. Okay, but this is not common. You won't see this. You'll appreciate this when you start your lab sessions. And then this, you'll see the pictures. Then we have fluorescence microscope. Basically, we expose it to ultraviolet, violet, or blue light. So as you can see this, it's a bright image. This is now what we call fluorescent light. It's going to expose it like this. Then we have staining. So staining is very, very important. It is actually the backbone of microbiology, how to identify these types of bacteria. So the three reasons: to increase visibility of the specimen, accentuates specific morphologic features, basically makes it clearer, and then it preserves the specimen. Okay, these are the reasons why we do staining. Then we have fixation. Sorry. Fixation means we are preserving them. From the word fixation, to fix it in a position. Okay. So it's the process by which organism is killed and firmly attached to a microscope slide. We have heat fixation and chemical fixation. For heat, we preserve overall morphology, but not internal structures because we're using heat. For chemical, we protect the fine cellular structure and morphology of the larger, more delicate organisms. Okay. So in any process, if you hear the word fixation, we're just preserving it. And then after fixing, we're going to see a procedure. You fix, and then you're able to observe it in a microscope. So staining, we have different types of stain. Okay, so many types of stains. I'm sure the common ones you've had is gram stain. Then for specific stains, depending on the bacteria, you'll hear ZN stain, which is used for TB. You'll hear Giemsa stain. You'll hear, it's called PAS, periodic acid sheaf stain. There's India ink stain, silver stain. There are very many types of stains. Okay. So depending on the agent used. So for example, so simple staining, a single staining agent is used. Okay. If you hear the word simple staining, a single agent staining is used. Okay. Basic dyes are frequently used, dyes with positive charges, e.g. crystal violet. Then we have something called negative staining. These ones are often used to visualize capsules surrounding the bacteria. So there are bacteria that we call them encapsulated. So we use a negative stain to visualize it. So capsules are colorless against a stained background. Can you see this colorless part. So that is how you'll know a bacteria is stained. There is a question they like asking. All the following bacteria are encapsulated except, or they'll ask, another way of asking, which is very high yield, they can ask you, all the following are positive with a Quelang reaction. Quelang reaction, let me type for you. So it was a way it was used back in the day. It's called Quelang reaction. So if it's positive Quelang reaction, we know that it is capsulated. If it's negative, it is not capsulated. Okay, I've written it on the chart. It's called Quelang. So if you see in your exam, Quelang reaction, just know those are the encapsulated bacteria. Then we have differential staining. So when you hear the word differential, know that these bacteria grow with different patterns. So here we are observing the pattern in which they grow. Okay, so divides microorganisms into groups based on their staining properties. So it can be gram stain, it can be acid-fast stain, etc. Okay, then gram stain. So this is the most important stain that we use in microbiology. Okay, it was the first line lab test. It is the first line actually that we use to classify or to identify the type of bacteria. So it divides bacteria into two groups based on differences in cell wall structure. Okay. So this is very high yield. You should know how the procedure is done. You should know the types of bacteria that are gram positive and that are gram negative. Okay. So I sent you a video. Did you watch the video on the gram stain? So for gram stain, okay, in this slide, I've started with crystal violet, but basically what happens before you expose it to crystal violet, you have to prepare the smear. Okay. So you have taken your sample. You usually take glass slide. So this is the glass slide. So I'm talking about what happens before we put the crystal violet. Okay. So we usually have a glass slide. You put maybe a drop of water, then you create a thin smear. Then after creating a thin smear, you put it on the air for it to dry. After drying is now when we do fixation. So the most common fixation is usually heat fixation. We pass it over a Bunsen burner flame around 2 to 3 times, and then the heat fixes the bacteria. So basically here, it's going to fix it, or it's going to adhere on the slide, and the bacteria will die. So there's no motion. After that, now we go to the primary stain, which is crystal violet. So it's usually purple in color. I don't know, is this purple or is it blue? Is it purple or blue? Or is it, they say men are colorblind. I don't know if it's true. But if you're going to see it in the lab, it's usually purplish, bluish dye, okay? Or navy blue, I don't know. And then we cover the smear. So this is the smear, I've already fixed it. We cover it with the crystal violet for 1 minute, okay? After that, so what do you think is the function of the crystal violet? What do you think is the function of the crystal violet? Okay, so for the crystal violet, it penetrates the cell wall. Okay, so it's usually known as the primary stain. So you'll hear primary stain, just know that is crystal violet. Sorry, let me take this. So I hope we've understood. So this is the primary stain. So after that, we rinse. Basically, using distilled water, we are removing the excess crystal violet. After that, we apply iodine. So it's called grams iodine, or you'll hear it's called mordant application. So what happens is we flood this slide with iodine solution. Okay. So also for 1 minute, the reason for putting iodine is because this iodine will form a complex with the crystal violet, okay, so that it retains its color. Okay, so in gram-positive bacteria, the color, this purple color is retained. Okay, then after that, we rinse again, but this time we are using a decolorizer, which is acetone. Can you see here? So we usually do for around 10-15 seconds until it's clear. Now, as you're going to notice here is gram-positive bacteria will appear purple because they will retain this iodine crystal violet complex. That's why they appear purple. But for gram-negative bacteria, they will lose the color. So they'll appear pink. So the reason for this is, as you're going to see, gram-positive bacteria have a very thick peptidoglycan layer. That's why this stain is going to remain. But for gram-negative bacteria, the peptidoglycan layer is very thin. So that's why the stain cannot be retained. And then we do counter staining. So the counter stain is methyl red or safranin, okay? There's sometimes you can use methyl red or safranin. So we do that for 30 seconds. So this is what is going to give the gram-negative bacteria to appear pink. Okay, since their cell walls have been decolorized. So you just put the counter stain, and now that is what will make the gram-negative to appear pink. After that, final rinse with water to remove this excess counter stain. And then we dry. After drying, we examine on a microscope. Okay, usually put it on a microscope. Then we put some oil. Okay, because that oil, it's usually called oil immersion, is going to give it a magnification. It's going to magnify it. And then you use, it depends, times, you can use times 40, times 100 magnification. Then the final thing is gram-positive bacteria will appear purple, gram-negative will appear pink. Okay, so this is a very, very high yield procedure. You can be told, explain the gram stain procedure, 10 marks. Okay, so this is very, very, very high yield. Okay, any questions on the gram stain procedure?
I have a question.
Yes, yes.
What's the use of the iodine solution?
So as we had said, iodine, the reason for iodine is it's going to form a complex with the crystal violet. Okay, we call it a mordant. So this complex is what is going to be retained in the cell wall of the gram-positive. Okay, is it okay? So you can say it acts as a mordant.
Okay.
Okay.
Thank you.
Sorry, this is a picture of a gram-negative rod. It's called E. coli. As you can see, they are rods and they are pink. So you'll know they're gram-negative. Okay, we go to acid-fast staining. This is also a high-yield type of staining. It is used in members of the genus Mycobacterium. So Mycobacterium tuberculosis and Mycobacterium leprae. So this one causes TB, leprae causes leprosy. Leprosy is not that common these days. I mean, I've never seen anyone with leprosy, but we still learn it. Okay, but they're in the same genus. So the reason we use acid fast and not gram stain in this is because the cell walls of these two bacteria have high lipid content. So they cannot stain with gram stain. Okay. So that is why we use this special acid. It's called acid fast staining. Okay. So I think we are done with the basics of microbiology. Any question?